Hey guys, hope you're well. We're going to talk about DNA today, more in detail. Um, okay. Okay. So let's get to what we're going to do. All right. Okay. So the general sequence of events, right, is DNA to mRNA to protein. Okay. So this is like a flow chart. Doesn't mean I'm converting DNA to RNA, RNA to protein. DNA is, think of DNA as an information molecule, right? It's the gene, right? So the gene is the code for the protein. It's a one-to-one relationship, right? So all the proteins we know out there, hemoglobin, collagen in your skin, keratin in your hair and nails, there's a gene for it, right? So in order to make the protein, you need to start with the information, the gene, and then you decode the information to get the protein. That's what we're going to be doing, okay? So if something goes wrong in the information, something's going to go wrong here. It's a one-to-one relationship. Gene, protein. Okay, mRNA is sort of like the middleman information molecule. Okay, so we know DNA has got two strands, double-stranded helix, right? And here are the general DNA to DNA hookup rules, right? So the nucleotide A is going to bind to T, G is going to bind to C, T is going to bind to A, C is going to bind to G. That's the way it's going to work. So let's take a sample gene. I'm going to call it gene X. Okay, so remember it's got two strands. DNA has got two strands. TG. Okay, so let's say this is a very small gene coding for a very small protein. Typically, genes are thousands of nucleotides long. Okay, so this is one strand of DNA. What would the bottom strand be? What would the next strand be?
T A C G G G T T T A C T.
Okay, good. T A C, very good. G G G T T T A C T. Does everybody see where she got that? Yes? No?
Yes.
Okay. Okay, so basically A with T, T with A, G with C, C with G. Okay, so this is my gene here. So in this gene is information to make protein X. This is called gene X. Yeah, sure. So remember, A hooks up with T hooks up with A with T with A, G with C, G with C, C with G. That's simple. One-to-one relationship. Does that make sense?
Yes.
Okay, great. All right, so the next step is sort of to make the middleman information molecule, that's RNA. Okay, now right next to every single gene out there is a box of nucleotides. So there's some ACGs and Ts in there that doesn't code for anything, but it's like a control box. Okay, this box is called the promoter. And we'll explain what the promoter is for in a second. Okay, so before I talk about RNA, how to make RNA, right? We're going to talk about RNA as a molecule. Okay, it's a little different than DNA. So RNA is one single-stranded. Okay, so we've got one strand in it. Two. So DNA's got two strands, RNA's got one strand. It's got the nucleotides A, G, C, and U, where U stands for the nucleotide uracil. There's no T, like in DNA. There's a U instead. And three, the sugar with these associated with these nucleotides is different. Okay. So the general hookup rules for DNA and RNA are this, right? If the DNA nucleotide are AGCT, the RNA, since there's no T, there's a U, G with C with G, T with A. Okay, those are the RNA hookup. It's the DNA. Okay, so single-stranded, no T, but a U, uracil. Okay, and these are how the nucleotides sort of hook up with each other. Okay, so the enzyme that's going to make RNA using the DNA is going to be RNA polymerase. Okay, that's the key enzyme. Okay, so this is what's going to happen, the sequence of events. RNA polymerase is going to land at this thing called the promoter. So think of the promoter as a landing pad for this enzyme. Okay, so it's not going to land like in the middle of the gene, it's going to land here. So it's going to land at the promoter. And what it's going to do is it's going to unzip the DNA strands, take them apart, and it's going to use the bottom strand as a template. So we've heard this term template before. What does it mean to have a Microsoft Word template? What does that mean? Everybody know what that means? So what does it mean to have a template outside of biology, a Microsoft Word template?
Like an outline or format?
Yeah, so it's like an outline or a formatable mold, right? So what this enzyme is going to do is it's going to land at the promoter. It's going to unzip the strands, and it's going to use the bottom strand as a template, an outline, to make mRNA. Okay, so let's assume it does that. Okay, so we have TAC, GGG, TTT, ACT. Okay, what would be the RNA strand it makes off of this DNA? Can somebody give me the
AUGCCC AAAUGA.
Yes, very good. Does everybody see where she got that?
Yes. Yes.
All right. T with A with U, there's no T, C with G, E with C. Yes?
Yes.
All right, great. So this is my mRNA. Okay, so mRNA, the M stands for messenger. Okay, now what does it mean outside of biology? What does it mean if I transcribe notes for somebody? What does it mean to transcribe notes for somebody? Yes? So I'm going to transcribe notes for you. What does that mean?
You're like writing them down.
Yeah, I'm copying them, right? So this process that we just did, it's called transcription. We're transcribing mRNA from DNA, using DNA as an outline or a template. Okay, so this process is called transcription. Okay, and since the DNA is in the nucleus, that's where this takes place, nucleus. Okay, so this process, making RNA from using DNA as a template, it's called transcription, and occurs in the nucleus. Yes?
Yes.
Okay. Now, we still have a bunch of letters here, nucleotides. The goal is to make protein. Okay, what are proteins made up of?
Amino acids.
Amino acids. Okay, so we still have yet to come up with amino acids. Not yet. Okay, so what we're going to do, okay, is first of all, we have to have a code. This is all about codes, right? So in here, there are codes for making proteins. So we need codes for amino acids. So we know the RNA nucleotides, A, G, C, U. Let's say one letter represented one amino acid, like a one-letter code. How many amino acids could I get here?
4.
4.
Okay, 4 is not going to work because we have 22 amino acids. How about a two-letter code? How many combos? AG, GA, CG, CG, CU, UC. How many combos are then possible?
8.
Close. It's 4 squared. 16. 16 is not enough either. Okay, so it's like armchair logic, right? So a 3-letter code, that's going to be enough, right? Then you get 64 different combos. That's more than enough to cover 20 to 22 amino acids. Okay, so what people did was they constructed like artificial RNAs, making out of just a triplet, like AUG. So they had AUG, 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 threw it into an artificial system, and they pumped out a protein just of one amino acid matching up to AUG. Then they did the same with, let's say, AAA, pumped out another amino acid, right, matching up to AAA, right? So they did this 64 different times using 64, and it's pretty tedious. And they came up with a chart, which I'm going to show you now, okay? So okay, so the chart they came up with is this over here. So this is called the chart of all the 64 3-letter codes. Now, since you have 20, at that time, 20 amino acids, 22 now, some amino acids like valine, this is the name of the amino acid, have more than one 3-letter code. 1, 2, 3, 4 3-letter codes. Some like leucine, another amino acid, has 1, 2, 3, 4, 5, 6 3-letter codes. Some like methionine have just one 3-letter code, AUG. Okay, these 3-letter codes, all these 3-letter codes are called codons. That's the name of these 3-letter codes. And 3 of them, stop codons, they don't code for anything, they code for stop signals, which will be clear when we talk about the next step. So now this chart, you don't have to memorize this chart, that's why it's here, right? Is universal on this planet for all living organisms. So what does that mean? It means CAU codes for a histidine in a bacteria, it codes for a histidine in a human or a plant, doesn't matter the organism. Okay, so on this planet, this is a universal chart almost. Okay, so how many total codons are there?
64.
Good, excellent. 64 total codons. Of those 64, one of them is AUG. That's called the start codon, which means all RNAs begin with AUG. Okay, so one start codon, AUG. And of those 64, 3 of them don't code for any amino acids. They're called stop codons. 3 stop codons.
So we only have one start codon and then 3 stop codons, right?
Correct.
Oh, perfect.
Okay, are we good so far, guys?
Yes.
Okay, so what's going to happen here is I'm going to introduce another type of RNA to you. I just talked about mRNA. What does the M stand for? Does anybody remember?
Messenger RNA?
Very good. Messenger RNA. Okay, so now I'm going to talk about a different type of RNA. I'm going to do it in red, and that's called transfer RNA. Or tRNA. Okay, it's a much smaller RNA than mRNA. It's like a hybrid molecule. In that, it looks like this. And one end of the molecule sort of hangs on like a hanger to an amino acid. And since it's an RNA, it's got nucleotides. The other end of it is a 3-letter code called an anticodon. So we have codon on the mRNA. We have anticodon on the tRNA. Okay. So the next step is we got this mRNA. Now we're going to use the information in the mRNA, the messenger RNA, to make protein. So what are we going to do? We're going to translate the message. So first we transcribe the message, and we're going to translate the message. So does anybody remember where in the cell we make proteins? Where amino acids put together?
Mitochondria?
That's the powerhouse. That's where energy is at. Right? So where are we going to put amino acids together? What are those little dots called?
The ribosomes?
Ribosomes. Remember the ribosomes, right?
Yeah.
RNA, ribosomes, right? That's where we're going to go. So we got to leave the nucleus, go into the cytoplasm, to those little dots, those little circles, little organelles, ribosomes. So what I'm going to talk about next is translation. Okay, any questions first? We're good? Yes?
Yeah.
Yes.
All right. So let's go back to this for a second. I'm going to erase some of this, right? You have two kinds of RNA, mRNA and the tRNA. Okay. So I'm going to go to the ribosome. This is translation. Here's my ribosome. B site, A site. Now, what do these mean? Just like you have an imaginary line to the center of the Earth called the equator, we have imaginary regions on the ribosome, which I'm going to call the P site, P for protein, and the A site, A for amino acid. They don't actually exist there. They're just regions of the ribosome. Okay, so this is the ribosome. The mRNA is going to come down here. AUG, so it's the mRNA, CCC, AAA, UGA. So my mRNA is at the ribosome. Now, we begin translation. The first tRNA is going to come in, matching up to AUG. So we will need to find out what AUG codes for. So let's go to the chart, codon chart. By the way, what do I type in into Google Images if I want this chart?
Codon chart?
Codon chart. Is that clear?
Yes.
Okay.
Yes.
So what does AUG match up to? Which amino acid?
Start codon.
Okay, that is the start codon. What's the amino acid associated with it?
Methionine.
Okay. Methionine. I'm going to abbreviate this MET. Okay. Let's keep it simple. Okay, so this is MET. So this tRNA is going to go into the ribosome and hook up with the mRNA. So this is a codon, anticodon, RNA to RNA hookup. This is an mRNA, this is a tRNA. They're both RNAs. A, U, U, A, G, C. Yes?
Yeah.
Okay. The next tRNA is going to come in. So I need to know what CCC is. So I'm going to go back to my chart. What's CCC?
Proline.
Proline. Okay, I'm going to abbreviate this PRO. Great. I'm going to go over there. PRO. So right now, we got two tRNAs hooked up to their codons. Okay? Now remember, this is a ribosome. It's an organelle. It's like a little ball. Inside here, there are enzymes. One particular enzyme inside this ribosome is called transferase. This is the important enzyme for translation. So this enzyme is going to do a couple of things with some help from other factors. But one thing it's going to do is it's going to remove this amino acid, which this tRNA was hanging on to. It's going to remove it from this tRNA, and it's going to bond it covalently, covalent bond, shared bond, to this proline. So here's the amino acid hanging on to the tRNA. I'm going to remove it on that enzyme. Here's my proline. I'm going to bond it to my proline. Okay, so it's going to look something like this. Look what's happening. This enzyme, transferase, remove this amino acid from this tRNA, so it's empty now, and bonded it to the amino acid being held by the other tRNA. So these two are now bonded. It's a covalent bond. We're good so far? Questions? Yes? No?
No.
Okay. Another thing that this enzyme is going to do, again, with a little bit of help from other proteins, is think of this ribosome as a ball and a bunch of railroad track of codons. This entire unit is going to move one codon over. So it's going to look something like this. This is 3. East side, east side. Okay, so look what's happened from here to here. Whatever was in the A side is now on the P side. We shifted one over. This empty tRNA gets kicked off. He's going to go pick up another amino acid. So he's kicked off. So there's nothing left on there. This side is now empty. What is AAA code for? So we're going to go look at AAA. What is AAA code for?
TTT. Oh, sorry.
No, go ahead. Which amino acid?
Lysine.
So I'm going to abbreviate this LYS. Here comes my third amino tRNA. Looking up here, LYS. So what's going to happen next? What's this enzyme going to do next?
It's gonna connect to MET and PRO.
Yeah, so these two are going to be removed from this tRNA and be bonded to this third amino acid here. Clear? I'm going to show you what it looks like. Questions on this? We good? Okay, so this is going to be 4. This side, this side. Okay. Notice what's happened here. I got 3 amino acids now bonded to each other. What's going to be the next step? By the way, what am I making here when I'm bonding amino acids together?
Proteins.
Protein. My protein. What's my next step? 5. What's going to be my next step? P side, A side. P for protein, A for amino acid. What's my next step?
You find the codon for UAG.
Well, what's going to happen to this whole complex?
Oh, it shifts.
Okay, I'm going to shift 1 over. Good. Okay, now we got to find out what UGA codes for. Go over here. What does UGA code for?
The stop codon?
Yeah. So basically, it's a stop signal. So some other factor is going to bind to it and kick off that chain. So that's where we stop. Right? This is going to be released as a protein. So my protein has how many amino acids on it? The one I just made.
4.
Sorry.
Well, if you count the stop codon.
No, stop codon is not amino acid, right?
Oh, so 3.
Okay. Stop codon is a signal to kick off the protein. Okay, so 3. So we started with my gene X. Now we got protein X made up of 3 amino acids. Okay, this in a nutshell is what translation is about. We translated the message. We good?
Yeah.
Questions, guys? Okay, so this is my gene X. This is my gene X that we started with. Like I said, I have a mistake here, and I have a mistake here. So let's say this particular gene gets hit by X-rays or ultraviolet rays, such that this A becomes removed and becomes a G, is replaced by a G. Would that make a difference? Yes? Guys, are you there?
I think it would make a difference.
Okay, yeah, it might not, because some amino acids have more than one codon, right? But in any case, I changed the DNA sequence, right? This is called a mutation. Mutation is any change in the DNA sequence. Okay, this particular mutation is called a substitution. Now, substitutions, it might have an effect, it might not. Bad substitutions like the one you have with normal hemoglobin versus sickle cell hemoglobin. So one, like a point mutation like that. But you can also have mutations like this. So let's say, sorry, this should have been, this first substitution should have been something like this. Let's say this is a G, this is a C. So basically the C is replaced by G. That's what I meant by substitution, not adding an extra one. But you could have that too. But let's say you have something like this, a complete deletion. This entire thing is gone. You still got to go in threes. Right? That's called a deletion. Okay? And if you can have a deletion, you can also have an addition. That's the first one I drew, actually. Okay, either way, you're going to change the sequence. Deletions and additions are the worst because they often lead to what I call premature stop codons. What does premature mean?
Before it's like meant to be.
Before it's meant to be, right? And so you're going to get a bigger protein or a smaller protein?
Smaller.
Smaller, usually non-functional protein. The mutations are important, right? So let's say in your skin, you have some genes or somewhere in your body that are involved in cell division, making cells divide, 2 into 4, 4 into 8, so forth. But let's say those genes that are involved in cell division, they get hit by X-rays, ultraviolet rays, or some other environmental factor, such that the gene gets mutated and the cell loses control of division. So now it doesn't stop dividing. What's that called? When your cell doesn't stop dividing.
Cancer?
Cancer. Exactly. So now you got a mutated gene leading to a cancerous cell. Cell is immortal, doesn't stop dividing. You get a clump of cells, it's called a tumor. Some of those cells break away and go to your heart, and we have problems. Make sense?
Yes.
Yeah.
Any questions on this, guys? Okay. So let's go back to the screen for a second. So here's the sequence of events that we just went over. Okay, what's this first part called? What's this called?
Transcription.
Transcription. What's this part called?
Translation.
Translation.
Very good. Okay, so transcribing the message, translating the message. Okay, now the so-called the vaccines that you're being injected with, they are mRNA, especially Pfizer and Moderna. Which protein do they code for? So they're injecting you with mRNA encased like a little nanoparticle into your skin, into your body. What does it code for? Your body's going to translate it. Your body cells are going to translate it into protein. You're going to use this code that they're injecting into you. But what's it coding for? This you should know, because it's a vaccine, my friends, and it's out there.
Is it the spike protein?
Exactly right. It's the spike protein coming from the coronavirus, right? So What's going to happen is they're injecting you with the mRNA coding for the spike protein. The mRNA is not going to hang around there a long time. It's going to be degraded. But your cells, in the meantime, will translate that code for the spike protein into the spike protein. So now your cells are going to have spike protein temporarily. Then you're going to make antibodies to that new protein, to that spike protein. Okay? Now you're ready. When the real virus comes and invades you, you got your weaponry, your weapons, your antibodies, all triggered for that spike protein. Does that make sense?
Yes.
Yes.
Okay. The other ones, the AstraZeneca and the Johnson & Johnson, they start at this level here, the DNA level. They use a harmless virus to sort of take it into your body. Here they're using like a lipid nanoparticle. But it's the same thing. The AstraZeneca and the Johnson & Johnson DNA code for the spike protein. Everything's about the spike protein. Clear?
Yes.
Yes.
Okay. Any questions on that? Okay. So this is the normal sequence of events. It's called the central dogma. Certain other organisms like HIV and even SARS are a little bit different. Okay, but HIV is special in a sense. It starts out as an RNA virus. So that RNA is a genetic material. And what it does is it converts that RNA to DNA, and then it goes the normal route. Okay, so in order to do this, and even SARS can't do this, it has a special enzyme called reverse transcriptase. Okay, we don't have this enzyme. They have this enzyme. That's why sometimes it's called a retrovirus. Converts RNA back to DNA, and then we go the normal route again. The reverse transcriptase is the enzyme. This is called reverse transcription. This is the reverse of normal transcription. So the first anti-HIV drugs blocked this enzyme back in the 80s and the 90s, and there was a drug called AZT, highly toxic. Now, of course, the drugs are much better. Okay? Questions?
No.
Okay. So we'll talk about one last thing, and we'll take a break, and then we'll start talking about DNA technology. People often study genetic systems in bacteria because they're easy. They're small genome, okay, especially E. coli. Okay, the way bacteria work is they have genes, just like us. They have a promoter for their gene. What's a promoter again? What was the function of the promoter?
Where the mRNAs.
So it's a landing pad for the RNA polymerase. Remember, that's the enzyme that makes RNA using DNA as a template. So it's a landing pad for the RNA polymerase. Yes? Are we good?
Yes.
Yes.
So in addition to the promoter, we have promoters for our gene too. They also have a separate sequence called the operator, which we'll talk about in a second. Operator. So they have a promoter and operators. These are like two control boxes. And then they have their normal genes. C, Y, A is an example. These are normal genes. So for us, we have one promoter, no operator, and one gene. There's one promoter, one gene. For them, they have promoter, operator, and usually 3 to 5 genes. Okay, this entire unit is called an operon. We don't have operons, bacteria have operons. And there are about 50 different kinds of bacterial operons. This particular one has a name because it's the most famous. This is called the lactose operon. And so the lactose operon is made for breaking down lactose. Remember, lactose is a sugar, disaccharide. So the genes here code for stuff, proteins that break down lactose as an energy source for the bacteria, specifically the Z gene. So these are the Z gene, the Y gene, and the A gene. Okay, the Z gene codes for an enzyme. They all code for proteins called galactosidase. The galactosidase breaks down lactose. Okay, so the RNA polymerase is going to land at the promoter, unzip the DNA, unzip this, and transcribe all 3 genes, Z, Y, and A. And then we can go through translation later. Okay, so here's a question. If you're the bacteria, let's say you're the E. coli, and there's no lactose around, do you want this operon on or off? If there's no lactose around, do you want this on or off?
Off.
You want it off, right? There's no lactose in it. So in that case, there's a protein called the repressor. So this is a protein, another protein called the repressor. It's going to go and sit on the operator. So just like the promoter was a landing pad for this enzyme, the repressor is a landing pad. I mean, the operator is a landing pad for the repressor. The repressor is going to sit over here. And let's say the repressor is over here, and the polymerase is over here. Okay, the polymerase can't move this way to transcribe because the repressor is blocking physically. Okay, now let's say there was lactose in the system. What do I need to do from the bacteria to get my polymerase to go? What do I need to do? So there is lactose. I want it on. What do I need to do from the bacteria? Guys?
You have to get rid of the repressor?
Exactly. Right. Okay. So lactose, what happens is lactose itself gets rid of the repressor, blocks out the repressor. So it helps its own breakdown. This is how the on-off system of this operon works. And there are 50 of them. One lactose operon. Clear?
Yes.
Yes.
Okay. So now I'm going to give you an intro, and then we'll take a break to gene engineering, because that's what we will be doing after for the next few lectures. Okay. So we need some tools to do this. One tool is something called a plasmid, which we've looked at before. Does anybody remember what a plasmid is?
Yeah, the space of singular DNA.
Yeah, well, bacteria have plasmids mainly, not humans or plants or anything. So bacteria is a small circular DNA that has antibiotic resistance genes. So it's a small circular DNA that has antibiotic resistance genes. Okay, so they found a lot of bacteria resistant to antibiotics, and it's not on their main DNA, it's on plasmid DNA. Let me show you a picture of it. If you just Google image a plasmid, you can see a small circular DNA. So here's the main bacterial DNA, here's the plasmid. It's actually a lot smaller than this. This is what it actually looks like. Circular DNA under an electron micrograph. Okay, we're going to use it for our purposes. Okay, so tool number 2, something called the restriction enzyme. Okay, so what is this enzyme? I'm going to draw this enzyme. So in the 60s, they found that bacteria have defense mechanisms against viruses that invade bacteria. If a virus invades a cell, it injects its genetic material inside the cell, whether it's RNA or DNA. What bacteria have are these special enzymes that can chop up this DNA, and those are called restriction enzymes. So this is another bacterial product. Okay, so one of the first, and there are thousands of these restriction enzymes, depending on the bacteria. One of the first ones to be discovered was this one here, EcoR1. This is the name of a restriction enzyme. It stands for E. coli restriction enzyme class 1. There are tons of others. But what's special about these enzymes is that they can scan the DNA, any DNA, your DNA, my DNA, viral DNA. And when they see their target sites, each one of these guys has a specific target site, they chop the DNA. So the target site for this enzyme is this. Notice it's the same backwards and forwards, GAATTC, GAATTC, right? That's kind of targets these enzymes recognize. So this enzyme, this particular enzyme will scan a huge length of DNA, and wherever it sees this target site, it's going to make cuts, usually here and here. So it's like splitting together a Lego piece. So you're going to get pieces like this. You're going to get a piece like this and a piece like this. Sort of like two Lego pieces coming apart. This end here is called a sticky end. So what we're going to do when we come back, we're going to see how we use these enzymes in the plasmid, right, to do some gene engineering. Okay, so that's tool number 2. Think of the restriction enzymes as sort of scissors. We're going to be cutting and pasting together, splicing together DNAs. Okay, the third tool comes from a virus called T4. It's called T4 DNA ligase. Okay, this is the glue. So if I can chop off pieces of DNA with restriction enzymes, I can glue them back with the ligase. The ligase is sort of like paste back together DNA. Okay, so we're going to use all 3 tools to do our gene engineering. And then we're going to start talking about other stuff like cloning and stuff like that. Good? So let's come back around 9:20 or so, and we'll pick it up. Yeah?
Yes.
All right, guys. See you in a bit. Okay, guys, hope you're back. Hello. Okay, okay, so we're gonna take some examples of gene engineering in action. Okay. One second. Okay. All right, so let's say we want to make, let's say, what do most diabetics need? What's the chemical most diabetics need?
Insulin?
Insulin, excellent, right. So where do you make your insulin in your body? Does anybody know? Which organ makes you your insulin?
Is it the pancreas?
You got it. Pancreas, right? It's insulin made by pancreas. So where does most of the world's insulin, how do the diabetics get their insulin? Where does that insulin come from? We're grinding up human pancreases? So what is that insulin coming from? Since you know it comes from pancreas.
Maybe animals?
Yeah, it's coming from beef or pork insulin, right? And so what we want is human insulin, right? But we don't want to grind up human pancreas. You can't do that, right? So we're going to show you a different way of making human insulin. This was actually done, right, in the 70s. Okay, so the first thing you need, okay, is to make a human insulin gene. Okay, so I'm not making the protein, I'm making the code. How do you make the code? You string together nucleotides. You get the sequence of the human insulin gene, and you can easily string together the nucleotides. So I'm making the code. Can't make the protein yet because it's got to fold in a particular way and everything. Okay, so here's my human insulin gene. Make human insulin gene. Okay, so here's my human insulin gene. It's got nucleotides in it, A, G, C's, and T's. It's my code. The second thing I'm going to do is I'm going to make some DNA adapters. Okay, what are these adapters? These adapters are little small pieces of DNA, so they're small. How small is my gene? Maybe like 20 nucleotides long. Okay, so these small adapters are like small pieces of DNA. And I'm going to make them so that they containing target site for restriction enzymes. So remember, restriction enzymes cut DNA at specific targets, right? Like EcoR1. That's the name of a restriction enzyme. Okay? So I'm making two kinds of DNA here. I'm making my human insulin gene, and I'm making these small pieces of DNA containing my target site for restriction enzymes, like GAATTC. So in here are some GAATTCs. Then I'm going to stick adapters onto gene, insulin gene. So here's my insulin gene. I'm gonna stick the adapters onto my insulin gene. So how do I stick them on? What enzyme am I going to use to stick them on? Using which enzyme? So what's my glue-based enzyme? Yeah,
Ligase.
ligase, exactly. So using T4 DNA ligase, very good. Okay, then I'm going to cut my ends, cut adapters with EcoR1. Okay, so that's going to leave me with something like this. Here's my human insulin gene. Here are my adapters. Notice I'm drawing them like a L shape. Okay, so remember what this L shape is, okay? So here's my restriction site. Okay, so the EcoR1 is going to cut here and here, and it's going to give me two pieces, right? Those are my two pieces. So notice these are L-shaped, right? That's what these are here. Same thing. Right, so these are these. Are we good?
Yes.
Okay. These are adapters, okay? Then, number 6, I'm going to draw 6 over here. Get a plasmid. Remember, plasmid is a small circular piece of DNA containing antibiotic resistance genes, and cut with EcoR1. So here's my plasmid. Okay, this plasmid is going to come with an antibiotic resistance gene. So this box over here is the penicillin, as an example, penicillin resistance gene. Okay, and what I'm going to do is I'm going to cut this open with EcoR1. And it's going to give me the same kind of sticky ends. Here's my penicillin resistance gene. All right, I'm going to go from here to here when I cut it. So here I got some ends that are staggered. I'm going to cut open plasmid. What I'm going to do is I'm going to take my human insulin gene with its sticky ends. I'm going to stick them into my plasmid. So stick human insulin gene into plasmid using the ligase. Okay, so now I'm going to get something like this. Okay, this is my new plasmid. Okay, so I sort of splice some new stuff on there, right? So what's this over here that I put in? What I stick in my new plasmid?
Human insulin gene,
Okay, and what's this red box over here? What gene is this?
penicillin resistance gene.
Okay, yeah, excellent. So we got two genes here, human insulin gene, penicillin resistance gene. So think of a plasmid as like a shuttle van. I'm going to use this shuttle van for something. Okay, so then what I'm going to do is find an E. coli, a bacterial strain, find me a bacterial strain with no plasmid. So essentially, I'm going to find me a naked bacterial strain that doesn't have any protection against antibiotics. It's got no plasmid in it. Okay? So here's my new strain of bacteria with no strain of plasmid. I'm going to mix my new plasmid that I made, new plasmid, plus E. coli strain with no plasmid, plus some calcium chloride. Okay, so this is what I'm going to have. That's step 11. I'm going to have a tube. Here's my bacterial strain without any plasmid. Here's my new plasmid that I just made. It's right now outside the bacteria. Mixing them together doesn't mean it's inside the bacteria. It means it's outside the bacteria. But what I want is, I want this new bacteria without any plasmid to take in my new plasmid into their body. So I'm going to get them to do that. The way I'm going to get them to do that is I'm going to put tube on ice for 10 minutes. So I'm going to chill my tube down.
What the fuck is he talking about?
Then put tube in hot water and the hot water is around, let's say, 108 degrees Fahrenheit for 30 seconds. So what am I doing here? I'm taking my bacteria from really cold ice to really hot water. I'm shocking it. It's like a shock treatment. And what that shock treatment does is opens up the bacterial pores, right? So these bacterial pores open up and this new plasmid comes into them. And does that make sense? It's like a shock treatment. Because I want the new plasmid, didn't have any plasmid before, to take in my new plasmid. Is that clear? Yes or no?
Yes.
Okay, that's not 100%. So how do I separate the guys that picked up my plasmid into their body versus the guys that didn't? So what does the new plasmid have on it? What's this gene?
Insulin gene,
What's this one here?
the penicillin gene, resistance gene.
Okay, so now, remember, these guys never had any plasmids before. Now they pick up a plasmid that gives them penicillin resistance. So what I'm going to do is I'm going to take my mixture here that I just shocked in plate, and then I'm going to a little petri dish on a penicillin plate. So anything that picked up the plasmid is going to survive because now it has penicillin resistance. Anything that is going to die. So I'm going to show you a picture of what that looks like when they put it on a plate. So these plates are like little petri dishes where they grow bacteria. So let's look at that. Okay, so let me show you a picture of what it looks like, the plasmids. Okay, so you see these petri dishes over here, right? Over here is some food for the bacteria, agar, it's like a gel. And inside here is some penicillin. So I'm going to plate my mixture, and these colonies that you see, these bacterial colonies, like this, these are the ones that survive. Okay, the ones that picked up my plasmid, because it now gives them penicillin resistance. The ones that didn't are dead. So you can screen for the ones that picked them up versus the guys that did not. Is that clear?
Yes.
Okay. So now what I'm going to do is I'm going to pick one of these colonies and grow them in some broth. And then I'm going to go to the bigger tank. I'm going to grow them finally in a big, huge fermenter that holds, 1,000 liters. Bacteria, they reproduce every 20 minutes. So in 2 hours, I'm going to have tons of bacteria in there. So now here's the important point. Remember, these bacteria are also doing transcription and translation. Okay? So what's this gene that they're transcribing and translating? What's this gene over here? What was this gene on the plasmid?
The insulin gene.
Insulin gene. Remember, this is a human insulin gene, right? Human insulin gene. So they're transcribing and translating my human insulin gene into human insulin protein. And so their little bodies are going to be packed with human insulin protein. I come back a few hours later, I crack open those billions of bacteria, and I got a lot of human insulin protein being made from it by these guys. Right? I purify it and I sell it on the market. So that spawned the biotech industry. It's the world's first biotech, 77, was Genentech. Right? Herb Boyer, biochemist at UCSF, is approached by Bob Swanson, who's a venture capitalist. Does anybody know what a venture capitalist is? This is outside of biology. This is economics. What's business? What's a VC do?
Is it like invest in companies?
Invest in companies, right? So mom and dad might not have enough money to fund you, but a VC has millions for a stake in your company. So they founded the world's first biotech, now multi-billion-dollar industry. It's bought by Hoffman and Roche, right? So what am I doing here? They're basically using the bacteria sort of factories. Here's my shuttle, my van, taking my package, the gene, human gene, into the bacteria, and the bacteria does the work for me. Okay, does the transcription and translation because they reproduce really fast. Use this plasmid, the stuff on the plasmid, and they make it for me. Okay, so the world's first biotech region became South San Francisco, then Cambridge, Massachusetts, then La Jolla. Those are the three main hubs. Okay, does that make sense?
Yes.
Questions, guys? Okay, that's what we're doing here, right? We can do this with any gene, not just human insulin. So for example, yeah, go ahead.
Sorry, for number 7, how does ligase become a factor? I was kind of confused on that step.
Okay, so ligase is a glue, right? So let's go back.
Thank you.
So remember I cut open my plasmid, right?
Yeah.
But how do I stick stuff into it? Like how do I stick the human insulin gene, that I had? How do I stick it into my plasmid?
Would you use the ligase?
Yeah.
Okay. Okay. Thank you.
You got it, right? Are we good, guys? Yes, no?
Yep.
Yes.
Does this make a little sense?
Yeah.
Okay, so this is new school, old school pharma ground up pancreases. New school uses gene engineering. Okay, so I could do this with any gene. So for example, most vaccines in the old days were inactivated, and still are to a big degree, inactivated viruses. They inject that into you, the whole virus. But sometimes the whole virus can become active. It's a 1 in 300,000 chance, right? So if I inject you with polio vaccine, which is the inactivated virus, if it becomes active in you, then you're going to develop polio. Right? You don't want that to happen, right? So let's say I look at hepatitis B. Hepatitis B. You guys know what that is? You heard of that? Hepatitis? Yes? No?
Yes.
So it causes target organ is the liver. So the hepatitis B virus, so let's say this is the virus, causes that disease. Now, maybe I don't want to inject the entire weakened virus into you as a vaccine. Maybe I look at it, it's got a lot of proteins surrounding some DNA. Maybe I use one of these proteins. Okay, the name of this protein is called surface antigen. Surface because it's on the surface of the virus, and it's a protein there. So it's one of the many proteins on top of that virus. So I'm going to use that. I'm going to get the gene for that. Here's my gene. Okay, I'm going to stick it on a plasmid. I'm going to get bacteria to make me that protein now instead of insulin. So now when I get enough of that protein, I clean it up, I inject that into you. Am I injecting the whole virus into you?
No.
No, I'm just injecting that protein into you, which comes with the virus, which was made by the bacteria for me. Okay, so that's a genetically engineered vaccine. The vaccines out there, I'm not using a plasmid. I'm using your cells to make the spike protein for me. I'm giving them the code, your cells make it. Okay, and you don't have to use a plasmid as a delivery system. You can use another virus, which is what Johnson & Johnson and AstraZeneca do, harmless virus. Any questions? We're good? Yes, no?
Yes.
All good.
Okay. What I want to talk about now is a little bit about cloning. And then the next time we're going to talk about DNA fingerprinting and forensics, where it's really important, right? And then a little about stem cells and CRISPR finally. CRISPR is like mind-blowing to me. CRISPR is where you can edit the genes, either in cells already, or I take your cells out, edit them, or I edit embryos, right? CRISPR is probably the most revolutionary thing in the last 100 years. Okay, and you'll see why. Okay, let's talk a little bit about cloning. What's cloning?
It's basically creating multiple of the same thing.
Yeah, it's making copies of yourself. It comes from the Greek word clone, which in the old days, clone means a twig, like a twig of a branch of a tree. They used to graft one plant onto another and you get a whole new plant. Okay, so that's where the term came from. So I don't know, I mean, this is before your time. You guys have heard of the Dolly the sheep experiment? Where they clone something? Yeah, well, okay, yeah, before your time, about 20 years ago. So I'm going to give you a human example. or what they did. Okay, so let's say I take a skin cell, one of you guys. Here's a skin cell. Your skin cell has 46 chromosomes in it. Every one of your body cells has 46 chromosomes, except your egg and sperm. Okay? So now what I'm going to do is, let's say, so the skin cell is one from one person. I'm going to take an egg cell from one of the ladies. Okay, so the egg cell has 23 chromosomes. Egg and sperm have 23. Come together, you get 46. So what I'm going to do next is I'm going to stick a micro needle, like a microscopic needle in there, into the cell under a microscope, and take out that nucleus. So now I'm going to have an empty egg cell, which is going to die if I don't do something, because I just removed the nucleus. So what I'm going to do next is I'm going to temporarily take my skin cell, an egg cell, empty egg cell, and sort of fuse them together with electricity, electrofuse them. So it's like a temporary process. I'm going to bring the two cells together. What's going to happen is the skin cell is going to serve as a donor cell. It's going to donate its nucleus to the empty egg cell. Not easy to do. Okay, now I'll get an egg cell with 46 chromosomes. So now it's ready to go. It can become an embryo. It's got the full set of chromosomes. I'm going to let it divide, and I'm going to implant that embryo back into the uterus from the young lady I got the egg cell from. 9 months later, you're going to have a clone of who? Where did the 46 come from?
Your skin.
Or some guy's skin cell, right? So you're going to have a clone of this person here 9 months later. In the sheep experiment, instead of a skin cell, they use a breast cell and they clone the sheep. Does everybody see what's happening here? Okay. What this is saying, this is important, is I use the skin cell to make a clone of that person. I need an egg cell and a donor cell. Every cell of your body, from your hair cells to your skin cells to your eye cells, has the information to make it. That's why it's so important. Is that clear?
Yes.
Does that make sense, guys? Yes, no?
Yeah.
Yes.
Okay. So why would people want to clone humans? What's the deal with cloning humans? What would one get out of cloning humans? By the way, other animals are being cloned. Let me show you. But what's the advantage of cloning humans? Why would you want to clone humans?
For like evolution or something.
Well, why would I want to make cloned human babies? Why would you want to clone them yourself? This is where bioethics comes in. Bioethics is big.
I guess to take your place in certain situations.
What's a baby? Or an embryo? Organs. Right? Organ farming. Got the same organs in you. Right? That's where bioethics comes in. Right? Otherwise, there's no real purpose, right? Does that make sense or not?
Yeah.
Yes.
Okay. So I'm going to show you like what other lists people have cloned, right? So, for example, so this is a list of cloned animals already. If you look over here, These are all the cloned animals, yeah? List of animals. Ferret, rat, camel, domestic cattle, dogs, goats, horses, mouse, monkeys, right? So these have all been cloned. Right now, they did an interesting experiment. Okay, let's see. Okay, so, and I'm going to show you some more gene engineering stuff in a second. This, can everybody see this?
Yeah.
This over here, yeah. Can you see this now?
Oh yeah, I see it.
Yes.
Yes.
So this is called the Spanish ibex, like a mountain goat. It roamed the area between Spain and France. Yeah, and they knew it was going extinct in the early 2000s. So what they did was they preserved a few cells before that, and they cloned it. So this is the first animal to be brought back from extinction, de-extinction. Right? It didn't live long because where you get your DNA from is important, right? And how old the clone is. But they proved it can be done. You can bring back an extinct life form. Okay, which is mind blowing. You never thought about de-extinction before, right? So the next thing people want to do is they want to. I'm going to show you another animal they want to bring back from extinction, and that is the thylacine, which I'm going to show you in a second. So does anybody know what a marsupial is? A kangaroo is a marsupial. What's a marsupial? Marsupials have pouches. Okay? So kangaroos is herbivorous, they eat plants, it's a marsupial. But you can also have marsupial carnivores, like the Tasmanian devil. Right? So, but the largest of the marsupial carnivores is this guy over here. This is called a thylacine. Okay? A thylacine, these are zoo images. They used to eat kangaroos. Okay, and they went extinct because human beings sort of drove them to extinction in the 30s. This is from like the 1930s. So they wanted to reclone this back. Very tough to do, but you need intact cells. Okay, but CRISPR might change all that. So another thing they want to do is clone the mammoth. Does anybody know what a mammoth is?
Yeah.
Yeah.
So what's a mammoth?
It's an extinct animal with like tusks.
Yeah, so it's a giant woolly elephant, right? They roam the Arctic. So mammoth 11. So you need intact DNA. So they've been finding intact DNA in Siberia, frozen mammoths, frozen baby mammoths. You need the DNA to be intact. So what you can do is the same thing, except you use an elephant egg. You implant this back into an elephant. And 20 months later, you should get a baby mammoth. Now, let's say the DNA is not intact. Well, that's where CRISPR comes in. We'll talk about that. Gene editing, right? You can edit the gene. So there's a company, called Colossal. Okay, so just got $15 million in funding. This is George Church, a very famous biologist. And so what they're going to do is they're going to use mammoth DNA, and any part that's missing, and a thing they can do is also they can edit elephant DNA into mammoth DNA. That's the beauty of it. So it's kind of like a mammoth elephant almost, right? So nowadays with CRISPR, you don't have to go find dinosaur DNA. You cannot have to find DNA. You can edit DNAs of something that's close to that animal, right? And so that's what they're going to do. And their goal is to make this, right? How to clone a mammoth, right? Does that make sense?
Yeah.
Yes.
Okay. So here, modify the genome of an elephant. Do something that look like a mammoth. That's what they want to do. Now you don't need intact DNA. So that's the beauty of this. Yeah? Okay, I'm going to show you some other stuff. And then we'll stop. So let's say I look at let's say this is a mouse egg. Let's say I'm looking at mice. That's a mouse egg. Here's a mouse sperm. Sperm is going to fertilize egg. Okay, so here's the mouse egg nucleus, here's the sperm nucleus. The two are going to merge. Happens with us too. But before they merge, I'm going to use a little microneedle again and inject the plasmid. This time my plasmid is going to contain the human growth hormone gene. So this is a foreign gene. It's a human growth hormone gene that I'm going to put into the mouse egg cell, sperm cell nucleus before they merge. Now mice reproduce really fast. So it's going to take a few generations of mice instead of make it into the sperm cell and egg cell line to get a mouse expressing this gene in every cell of its body. So what kind of mouse am I going to get? Say I get a mouse after 2 generations that expresses the human growth hormone gene in every single cell of its body. Okay, that kind of animal is, by the way, it's called a transgenic. Transgenic means it's expressing a foreign gene. Foreign because this is a human growth hormone gene. So what kind of mouse am I going to get? Yes? Take a guess. So what would it look like? So I'm going to express this gene in every cell of the body. What would it look like?
Like a human?
No. What's the gene? What's the gene here?
The human growth hormone gene?
Okay. So what would the mouse, okay, now it's making the human growth hormone protein. What would it look like? It's going to still be a mouse. What's going to look like? Okay, so I want you guys to think logically here, right? Growth hormone gene. What does it do?
Would it just be a huge mouse since they got the growth?
Does that make sense to you or me? Yes?
Yeah.
Yes.
Big mouse. Okay, so it would look something like this. Control mouse, growth hormone mouse. Yes? Okay, these are called transgenic, foreign gene. Okay, now that's not the only gene. So for example, okay, human growth hormone gene is one gene. Another gene is something called the green fluorescent protein, or GFP. This comes from a jellyfish. If I shine ultraviolet light, it glows green. It's a good way of tracking other proteins because I can tag any gene I want with this gene, and I can shine a light once the protein is made, and I can follow my protein in the cell. But I can also put it into mice, just like I did with the human growth hormone gene. So what you're going to get is something like this. I shine UV light on the mouse, GFP mouse, normal mouse. Yeah? So I can make these transgenic animals. I can stick foreign genes in them. Now, making big mice or glow-in-the-dark mice is not necessarily a useful task, right? But what I can do is I can also do it with fish. So transgenic salmon, normal salmon, growth hormone gene salmon. Now we're talking about food here. So different ballgame. Make sense? right? So these are your genetically modified foods. You can do it with plants too, right? Insect-resistant plants, growth hormone salmon. Yeah.
Yes.
Any questions? Does this make sense?
Yes.
Yes.
Okay, so what we're going to do next time is we're going to talk about DNA forensics and DNA fingerprinting, and we'll try to do what's in the kit, something in the lab kit, right? Using that for the DNA part. Any questions? We're alive? Yes? No?
No questions.
Okay, so remember to take your quiz on enzymes between 11 and 12. Yes?
Yeah.
Yes.
Okay, and we'll pick it up. We're going to do metabolism today. The data is all there. It's a simple lab. I'll tell you what I want in the lab report. Yeah?
Yes.
Okay, so we'll see you back here in a couple. Yeah?
Thank you, Professor.
All right, guys.
Thank you.
Thank
No.
you.
Recording stopped.